Thank you everyone for joining our webinar today. So my name is Ron Myers. I'm the Director of Editorial at JoVE. So today we're joined by Dayan Knox from the University of Delaware. So he published a paper with us back in 2019, and this was very well received. So we wanted to, kind of ask Dayan to give us some updates on the field. So today he's going to be talking about near-infrared fluorochromes and their use in neuroscience research. So I will leave it to you, Dan. Thank you.
Thank you, Ronald, for the warm welcome. I appreciate it. All right, so this is meant to be like a more informal, excuse me, more informal discussion about near-infrared immunohistochemistry, like how you use it. More details are in the article that Ronald mentioned, and through some of the publications that we have that you can see on PubMed or on my research website, you can see how we use the technology. So not meant to be like manuscript formality, more to be presentation. Here are some ideas of what you can do with this that maybe doesn't seem as obvious perhaps as it is to me. And then a question and answer session about anything to do with the technology. Okay, so I'll start off with the title: using near-infrared technology in neuroscience research. So I'll have some people who use current members of the lab and a former member of the lab, Brianna Schultz. She did a lot of work in one particular study, helping to utilize this technique. And Michelle Barth and Sushma Boyati are undergrads at the University of Delaware currently, but they have been a tremendous help to the lab. In fact, my lab is primarily undergraduates, and it's worked out really well. Okay, so that's a lot of work. Let me change this to a pointer. So I'm going to say some stuff here that's pretty obvious. What's immunohistochemistry? I was thinking, should I do a history about it? I didn't know if I could do it well because it's been used for a long time. Okay, it uses parameters of the immune system to characterize histological features of tissue. Now, if you want to look at features of a cell, like let's say the endoplasmic reticulum versus a Golgi apparatus, technically speaking, it's immunocytochemistry. I just thought I'd bring that up. I don't know if it really makes that much of a difference. And it utilizes the fact that our immune system has these cells that produce antibodies that recognize foreign proteins, typically expressed on the surface of foreign cells, even foreign organisms. And then it's a way of tagging them, and your T cells or macrophages will then destroy those cells. Now, that's great. That's why we could survive a whole host of insults that are thrown at us every single day. But you can also use that antibodies to look at proteins in any tissue. So immunohistochemistry, because I'm a neuroscientist, I'm going to talk about it from a neuroscience perspective, but it is used in almost every single biological sciences, life sciences field. Okay, the antibodies can originate from a single source. So one type of B cell, in that case, it's a monoclonal, or a variety of sources, different types of B cells. Sometimes it's in an animal, so you have a rabbit, so a mouse, and typically if you want to recognize protein from a rat, you get a rabbit antibody that recognizes a rat. But you can have mouse antibodies that recognize mouse proteins too. And then you have a lot of antibodies that are generated from cell lines. So it's not like animal was used to generate it. It's literally a culture of cells that generates the antibodies. Okay, but for this all to work, I mean, in your body, when chat. So currently in your body, if your B cell produces an antibody, that's either to inactivate some kind of foreign cell, target antigen for destruction, essentially. Now, if you want to see that reaction, that one of those antibodies have to have a tag, and then you could view the antigen antibody complex under a microscope or some kind of device. Okay, so hopefully it'll be more clear if I can get this to advance. As I go along, I'll show it in a cartoon. So typically in neuroscience research, you use IHC to measure protein levels in the brain. Here's some examples. IgGs is immediate early genes. Essentially, that's really all you're ever doing with immunohistochemistry, right? You're measuring protein levels. But because, for example, IgGs increase with neural activity, so if a neuron becomes really active, it upregulates these parts of AP1, CFOS, and CJUN, and you can use that as a proxy of neural activity. It's been used as a proxy of neural activity by, I'm pretty sure, hundreds of scientists all over the world. So you're measuring a protein, CFOS, CJUN, ARC, but really what you're measuring is neural activity throughout the brain. And if you have an antibody that recognizes protein and irrespective of whether it's modulated, AKT, for example, but then you have another antibody that only recognizes a protein when it's phosphorylated, PAKT, for example, then if you can measure these two proteins in the same tissue, you can look at signaling. You could look at how AKT phosphorylation changes with some kind of event, let's say stress. So in the end, all you ever, and one other example, this has been used, I don't know how well, but if you have a way of using immunohistochemistry to separate this cytoplasm and the nucleus of cells that you view, and then you can see your immunoprecipitates with any nucleus or cytoplasm, then you could look at things like receptor internalization, which is really important for steroid hormone receptor function. So no matter what you use immunohistochemistry for, it's always just measuring a protein. But if you measure the right protein, or measure two different forms of a protein, or measure protein in different compartments, you could not just look at expression of a protein. You can look at neural activity, molecular signaling, and then kind of cellular processes critical to neuronal function, or in case of glucocorticoid receptor internalization, stress reactivity. All right, so it's much easier to show in a picture. In all reactions, it's always the antigen, right? You're looking for a protein, and then you have a monoclonal antibody, a primary antibody that recognizes this, and then a secondary antibody with a label that recognizes the primary antibody. So antigen, let's say it's a glucocorticoid receptor, since I started with that, you would need a rabbit primary antibody that recognizes a rat glucocorticoid receptor. And then typically you use in a goat, could be a donkey, sometimes mouse, you use a IgG. So it's an antibody that recognizes rabbit antibodies that are made in another animal. And you can attach a label to that antibody. And the label can be HRP, biotin, or fluorochrome. And that will allow you to see this kind of big complex of proteins. But in the end, really all you want to see is this one protein here. Now, you can also use primary antibodies that have a direct label attached to them. So you don't need a secondary antibody. I've tried that a couple of times, never got it to work. But it's there, it's available, and people have got it to work and have used it. Okay, so one detection, if you have biotin or HRP attached here, usually you can make a reaction with diaminobenzidine, and then you get a precipitate that stays within a cell, it doesn't burst the cell body, and you get nice visualization of your protein product. So this is this reaction using biotin-related secondary antibody that recognizes choline acetyltransferase. And this is an area of the basal forebrain preoptic area. I was doing some lesions with a toxin. I wanted to show that I didn't target this area. So this is a lesion rat, this is a sham rat, this is about 40x. And the black dots that you see here, depending on what screen you're looking at, are neurons that express cholinestyltransferase. Now, only cholinergic neurons in the basal forebrain express cholinestyltransferase. That's the enzyme that synthesizes acetylcholine. And so you can make the argument that actually you don't really have to make the argument. You're watching cholinergic neurons here. And this is a light reaction. So DAB produces a precipitate. It looks black. If you do your reaction well, you have a decent background. And then you could use a light microscope. And the better your light microscope is, the better images that you could take. So this is when I was a grad student. The microscopes now are infinitely better, and the better ones are cheaper for light microscopes. So the label, instead of being having biotin or HRP attached, it could also have a fluorochrome, and that's just a protein or some chemical that when you stimulate it with one type of light, it's going to emit another type of light. And then you can use the light to see this whole antigen, primary antibody, secondary antibody reaction. So this is, again, basal forebrain. A lot of the research I've done has focused on the basal forebrain. And here we are looking at different proteins in the same cell. So this is a tracer. This is not really IHC, but this is CFOS. And this is, again, chat. So CFOS is a measure of neural activity. And we were using this tracer to tag cholinergic neurons that projected to the ventral hippocampus, so brain region. And so you can see you have kind of three colors here. This neuron is an active neuron that expresses the tracer, so it projected to the ventral hippocampus, and also has chat, so it's cholinergic in nature. You could see some other neurons that they're not, I didn't show them well here, that express a tracer but don't have chat. And then you can see they're definitely active neurons that neither project to the ventral hippocampus or have chat. So in addition to just labeling a neuron, when you use fluorochromes, it gives you a different level of differentiation. It gives you more options into the things that you can do with immunohistochemistry. All right, so with any technique in science, there's always pros and cons. There's always issues that you have to deal with. So it doesn't mean that what I'm about to say means IHC is something, it will be a technique that will be used as long as neuroscience is around. But there's always issues with any technique. So some of the pros with IHC when you use it in light microscopy. So there's no fluorochromes, you're shining a bright light, and you're hoping that you have enough of a dark precipitate so that you can see contrast between these dark things and a light background to characterize the neurons you label. Okay, so pros. Seeing focused neurons is possible. You can get really good contrast images. And these days we have an EVOS microscope. You can do a tile and stitch with a light microscope and get like a 40x image across an entire brain region that's really crisp, really sharp, and really easy to analyze. And this is good for seeing neurons in a single plane. Now, I can't really see myself, but in a 30 micron or 50 micron tissue slice, typically what you see is a top, a middle, and a bottom layer. And it's really good at focusing in one layer, not really capturing every single layer. And that's unfortunate because you do have kind of neurons in these different planes. Okay. So cons, typically you just see a cell body. It's hard to see anything. Like people have used immunohistochemistry with DAB and light microscopy to show dendrites and cell bodies. I'm not saying it can't be done. It's not often done. It's not easy. It's not very reliable, at least the times that I've tried to do it. You can label multiple proteins. There are companies that sell kits where you can get something that looks like dark black, something that looks brown, something that looks purple. And those colors merge really fast when you look at a bunch of neurons under a light microscope. But it is there. People do use it. It does get a little difficult, though, to distinguish, is this dark brown or kind of a light black, or is this kind of a really dark purple versus a kind of medium black? But so there's some differentiation, but If you want to differentiate, people typically use immunofluorescence, all right, immunohistochemistry, using fluorochromes, and there are lots of pros, right? You can see focused neurons in any plane. So if you have a 30 micron slice, and you kind of have one cell body here and another here and one here, you can see all those three cell bodies using a confocal microscope. Now, they're not easy to use, and it does take a learning curve, and typically you need at least one person. It's better to have a team who knows how to use it really well, who can then teach a bunch of other people who are interested in using it, how to use it. Now, the reason why you have to use a confocal microscope is because light scatters in your brain, right? You have a cell membrane that's scattering light, and then you have myelin around the axons that's scattering light as well. And so it's great to have light coming out, but when that light comes out and it gets refracted, and you just look at that raw image, that's wide field microscopy, everything is going to look blurry. If you want it to look crisp, like it's a light microscopy, then you need this confocal microscope. So it's not cheap. It's not the easiest thing to use. More and more labs use it either through a core or they just acquire the expertise. But that's what you would need to make seeing neurons in focus in many planes, to be very specific, to see features of neurons that are not restricted to the soma, to see multiple proteins. So that's a huge advantage. The image I showed with chat, CFOS and a tracer, you can see that image with relative clarity because I used a confocal microscope to do it. So fluoro, if you want to say IFC, but let's just say IHC with fluorochromes is a huge advantage, but it's always with the caveat that you need a confocal microscope or some kind of 2-photon microscope to deal with the issue that the light that's emitting from these fluorochromes will scatter in your brain, and you have to take that into consideration. Okay, so you can visualize multiple. 3 is the most that I've ever done, but it's not 3 where you're saying, hey, is this dark blue or is this really light black? It will be really clear, the color scheme. Okay, you can see neurons in multiple planes, but most people, they take what you have is a neuron that, and the cell bodies in like here. So it's in the top plane, in the middle plane, and the third plane, and you create a Z stack and you smush it all together and you see a really super clear image of many different things. People have shown microglia kind of pruning dendrites of neurons using this IHC, again, just looking at proteins. So very powerful technique. Now there's always some cons. Everything I just set up here, really, a nice powerful microscope, a nice powerful confocal microscope, or a 2-photon microscope. The concentrations of the antibodies that you need to get this to work is considerably higher. So for the DAB reactions with light microscopy, I typically use 1:2000, 1:1000, 1:500 for me would be kind of getting high. But for the IHC, 1:100 is not unusual. That's kind of typical. So, and they're not, the antibodies themselves are not cheap. So it does cost a little more. In addition to the hardware, the reagents that you need tend to cost a little more as well. And then, of course, something that I forgot to mention, because you're just measuring protein in tissue, you're measuring something that has already happened. So immunohistochemistry, let's say neuroscience, you have an animal, does a task, and you want to measure neural activity, you have to euthanize the animal, perfuse the brain or extract the brain quickly and freeze it, and then process it. So you always measure something that has happened already. It's not like single unit electrophys where you can measure something that's ongoing. Right now, you have to quantify all this, right? You did your reaction, you have your proteins, how do you quantify it? So I created a number of examples, and the most common technique with IHC is counting cells. So let's say you want to measure IgG levels after stress in a particular brain region. Let's just call it the amygdala. And then, you want to see, does stress increase neural activity? Now, the answer to that already is yes. Okay, so you can imagine these are pools of neurons, each circle in the amygdala. Now, some neurons are going to be active, and so they're going to express, some pools will express your IEG. And then after stress, the number of pools of neurons that express an IEG increase, right? You still have your baseline activity though. And typically these are two independent groups of animals. One animals that had no stress. This is what you could expect to see in theory. And schematically, let's say this is what happens. And then another group of animals, you stress them and you see all this increase in IEG expression. Then you can then plot on a graph. Essentially, you're counting cells. You're saying the number of cells that express CFOS goes up with stress. This is typically how you characterize IEG levels using immunocytochemistry in many neuroscience experiments. All right, a couple of things to keep in mind is cell counting, non-biased. So image J can count cells. It works like, I don't want to say perfect, but it works really well in cell culture with small numbers of cells, in tissue where there's good spacing between cells. It can work in the brain, but in the brain, it's a small amount of units, neurons, that's scattered in a relatively large area. So to capture that area, you have to tile and stitch a bunch of images. Then you have to keep track of what you're counting. That's not to say that you couldn't do it in a non-biased way, but a lot of care has to be taken that the cell counting that you're doing is non-biased. Okay, is cell counting inflated? Now, if you're counting cells in the amygdala, let's just pick a region, the central nucleus, any cell, all the GABAergic cells that express CFOS after stress, it's a lot of cells. You're going to get a really high number. As a result, your error bars are going to be super low. And then you are going to have to at least think about, is some of the differences I see being driven by the fact that I have these super large numbers. And so I have this really low N driven by these super large numbers. And, is that artifact? Is that cell counted inflated? I kind of made that up. But really, it's just this broader class of statistical things that you have to take into consideration. But you can usually take these into consideration, but you have to. All right, let's say you have another experiment where you want to measure glucocorticoid receptor levels after stress. Now, glucocorticoid receptors bind with glucocorticoids, go from the cytoplasm to the nucleus, and modulate expression of genes. That's typical. But let's say there's a stress condition where glucocorticoid receptors become upregulated. Now, here you have a bit of a problem because unlike IEGs, where there's very low levels or supposedly very low levels, when there's baseline activity, there's nothing really much going on, there's always glucocorticoid receptor. There's always going to be receptor in every cell in the brain. It's widely expressed. And so after stress, this is a theoretical situation that we are going to assume is true. You get an increase in glucocorticoid receptors, and you're trying to pick up that increase by counting cells, right, that express glucocorticoids. What are you really counting, right? All the cells express glucocorticoid receptors. And this is particularly acute for like DAB reactions. The only way you can get an increase is that all the cells that express a fair amount of glucocorticoid receptors express more. How are you going to capture that by counting cells when they all express a receptor to begin with? So a similar argument would apply for AMPA receptors. All neurons express them. NMDAR receptors, all neurons express them. Can you realistically do a cell counting technique to capture this relationship? So for the glucocorticoid receptor, many researchers, myself included, use Western blot, right? Because then you get a number that tells you in, let's say, the hippocampus between no stress and stress groups, how much glucocorticoid receptor you have. That's nice and convenient. Now, could you bring that number to immunohistochemistry? And I guess that's what the NearInfrared IHC thing is about. Okay, it's not a solution to everything, and it too has its own set of problems, but it's something to think about, I guess. So why use it? So the brain can be tough to study. In addition to scattering light, it also has its own autofluorescence. It's so bad or good, depending on how you want to look at it, that the autofluorescence, it's not just this brightness that you see. If you've ever looked at a brain slice under a fluorescence microscope, you know what I'm talking about. You can see neurons that look like they express GFP. There's no treatment. There's no genetic manipulation to get GFP in those neurons. They autofluoresce with such specificity that it looks like they're expressing GFP. Okay, so GFP, I think, is like 488, I believe, nanometers in terms of electromagnetic radiation. So near-infrared light is kind of somewhat removed from that autofluorescence range. So let me try to show what I mean. So this is the range that a lot of fluorescent tagged proteins are in. Anything over 800, but in neuroscience research, really anything over 680. So kind of here is considered near infrared, and that's great because this is white matter autofluorescence. As you get to 630, that autofluorescence kind of disappears. So that's typically a good reason, a reason why you would want to use near infrared antibodies, because they kind of avoid a lot of autofluorescence noise in the brain. If you're going to use, this is less about why. Here's a scanner for confocal, and there are new confocal microscopes that have cameras that can detect near-infrared antibodies. They're not widely available to the best of my knowledge. But for the near-infrared IHC, this is widely available. You can go buy it. This is Lycor. There are other companies that sell it, and you would put your slide down and you scan it. So here, kind of put this down, you scan over your image, see what you want to see, and then you set that confocal thing, and you kind of do a tile stitch to capture the part of the image that you want to analyze. Whereas this is capturing the entire image, and then you use a software program to analyze the part of the image that you would like to analyze. So 21 micron resolution, there's no magnification in scanners like this. They're just really, they have a really high resolution. So it's really small pixels of space that they're capturing when they acquire the image. So what do these images look like? So this is an example. We always do a validation because we know this is relatively new-ish. So this is first and secondary antibody for CFOS, secondary antibody alone. This is no antibody. You kind of have to show you can see something like this to know that what you're looking at is actually this antigen, primary, secondary antibody reaction. Let's see what's here next. So you can do a double label as well. Most scanners have 2 channels. And here we were looking at GLUR1 subunits in the amygdala, NR2A, so NMDA receptors, and then we did a merge. And so you could kind of look at ratios of GLUR1 to NR2A within the amygdala. And again, showing that secondary antibody, there's background noise here that starts and no in this channel, and then no antibody. And just to be clear, these are pseudo-colored. It's not really green. 800 nanometers of light. I'm not really, it will look reddish. It kind of look black and white actually to our eyes. All right, this is not something that we've used. So this is the ventral hippocampus, the cellular layer. You can put a line across it, and then you kind of get this normalized signal. And instead of measuring the mean intensity within an area that has signal, you can measure normalized intensity within an area that has signal. So we haven't used this as much. We've used this. Okay, so these are what the images look like. And this is a 1x, but if you could zoom into this with and get a lot of clarity, so it doesn't lose resolution as you zoom. You have to zoom a lot before you begin to lose like before the image gets really blurry. Okay, so why use NearInfrared? Because I mean, just like cell counting can pick up this change, you know, NearInfrared immunochemistry can pick up this change as well, right? If you're just measuring protein across this area at 21 micron resolution, that's about the size of a soma of the neuron, then you should be able to pick up something like this. But it can also measure something like this. If you measure protein in this area, you should be able to pick up the baseline activity as well as the increase that occurred with stress. Well, these are some of the reasons why you might want to, you can use near-infrared IHC. All right, so this is some of the things that we used it for. We used it to look at neural activity and functional connectivity patterns. That's something I was really interested in, and how those things change with traumatic stress, especially functional connectivity, because we are taking a number that the machine puts out for us. It really works well with a simple statistical approach factor analysis. It allowed us to group similar correlated levels of CFOS and CJUN expression. Those results matched up pretty well with the literature and allowed us to, make some inferential statements about how stress was impacting connectivity between different brain regions at different time points, like extinction training, extinction testing, fair conditioning. There's a limit to how you can use this, but that's something that you can use it for. All right, what our most recent publication is using it to measure fair learning-induced changes in AMPA-Rs and NMDAR expression within the FAIR circuit. So fair memories, at least a way, really established way that it's formed, is that it's synaptic, and you have sensory input onto amygdala neurons that initially only have NMDA receptors. Now, as that input becomes strong, right, sensitized, so to speak, then you're going to get active AMPA receptors, and the ratio of AMPA currents to NMDA currents increase. The next time that neuron gets active, then it's easier to stimulate an amygdala neuron. So an increase in AMPA to NMDA currents typically accompanies things like increases in fair learning and memory. So we wanted to look at broad changes, not in a neuron or a synapse, but across a region, in AMPA receptors, NMDR receptors, in many different brain regions, right? Not just the amygdala and the ventral hippocampus and the MPFC, midline thalamic nuclei, and then see if those changes are sensitive to protein synthesis. And that's what we did here. So this is a thing that you can do. There are others that we would like to do. But one of the things we looked at is mu-opioid receptor expression. This is typically done with Western blot, but take, for example, the amygdala. It's not just one homogeneous brain region. It's subdivided into a lot of nuclei. If you wanted to look at central nucleus versus lateral versus basal, medial, how does mu-opioid receptor expression change with stress? It's hard to use Western blot, right? You have to use a technique that will allow you to have that spatial resolution to look at those receptors in those brain regions individually. This is a technique that you can use. This is what we did. One of the things we did in this publication. All right, no technique is perfect, neither is this. One of the most common critiques I get, and it's justified, you can't see a neuron, right? You have this 21 micron resolution, it's high, but there's no magnification. And so, yes, you're looking at protein expression, but it would be great if you could, in addition to looking at protein expression, zoom in and then look into the individual neurons. If there's a way to do it using this, I'm not aware of it. One of the things that's kind of weird is that IHC that works with DAB and fluorochromes, when you use a microscope, it doesn't always work with the scanners. So glucocorticoid receptors is an example. So I still use Western blot to detect glucocorticoid receptors. Estrogen receptors is another example where it can work with DAB and fluorochromes. It definitely doesn't work when you try near-infrared IHC. I'm sure there's a reason why, and I'm sure it can be figured out. But once we can't get that validation assay to work, usually we just skip over. And then sometimes the validation assay works, but it doesn't work well enough to do a quantitative assay. And so then, kind of just, it's over. Most scanners offer 2 near-infrared channels. I guess, 3 would be great. 4 would be even better. And scanners are very expensive. The newer ones especially are really kind of cost prohibitive. I think a university would have a problem, or medical school, if there were 5 researchers that were using, even if it was grant money or startup money, to buy 5 different scanners. They are expensive, and just the price kind of forces an institution to put them in a core, as opposed to an individual lab having them. Right. So acknowledgements time, I went a little over than I planned. These are some of the members in the lab. Negan Mohammed Mirzai, who's my PhD student. She did, sorry? Oh, somebody worked with the traumatic stress. Brianna Schultz, former lab manager, an undergrad student who did a lot of the work. Brianna, Bailey, and Abby were first authors on that AMPA-R and MDAR paper we published in Learning and Memory. Sushma, Michelle, and Donna, who spent a lot of time working on the glucocorticoid receptor assays. It looked like it was going to work. It just didn't. There's part of the frustration with using this particular technique. And Matt, my former lab technician, who's really instrumental in just helping all of this research as well. Last but not least, Jeffrey Kaplan, he's director of microscopy core at the University of Delaware. His conversations with me have been great. Funding, and now references, it was only 3, and now questions.
Thank you for the great presentation. Sorry, I was on mute there. So yeah, if anyone has any questions, please add them in the Q&A or the chat.
Okay, so I see one. The near-infrared antibody dilution used to perform the techniques are the same one used with DAB or fluorescence. So fluorescence, when I've done it, the antibody dilutions are higher considerably. I would say the near-infrared, they're somewhere in between. DAB is definitely, in terms of reagents, if you're talking about money, economy, DAB will give you the best bang for your buck. The primary antibody and secondary antibody dilutions are the best. Near-infrared, you have to increase it a little bit. Actually, not a little bit. You do have to increase it. And then for immunofluorescence, you really have to increase the concentration to get really good images, at least when I've been using it. These days you can use Alexa Fluor. There's 2 companies that I know that make these machines, Azure and Lycor, and they both have fluorescent detectors and lasers in their machines. I've seen like Western blots. So I'll just answer the question. Yes, you can. Not the scanner that I have, but the newer scanners that are out there, yes. So, okay, the first part. Yeah, the alternative. So there's a limit to what I can say, because I have to do my best not to try to promote or not promote a company, right? Just given my particular precarious situation. So I would say there are alternatives. You can search them. They're available. I would even go so far as to say widely available near-infrared secondary antibodies. You don't have to purchase them from Lycor alone. I've only ever purchased them from Lycor, and sometimes I just like to keep things the same, even if I pay a little more money. It just makes my life feel a little easier. But you can purchase near-infrared secondary antibodies at the exact same wavelength emission as the Lycor antibodies from a different company. So the answer to that is yes. Fluorescent conjugated primary. So we did try one time where we had a primary antibody that had a near-infrared fluorochrome attached directly to it. to try to save time. It didn't work. Other people get it to work in DAB reactions and immunofluorescence. So I'm pretty sure that it would work. It's just that we tried it once, it didn't work, and we just stopped trying it. You can get 780 and which is they call 800 CW is 780 and 680 RD, which it's still 680 nanometers emission. Both of them you can find from companies that are not LICO. So the last question was, what wavelength of antibodies secondary antibodies can you purchase that fluoresce in the near infrared spectrum can be purchased from a company that's not LICO? So I said 780 nanometers and 680 nanometers. So both antibodies are LICO cells. You can find comparable products from other companies.
That may be it for the questions. I don't see any others. Let me check. Yeah, I think that's it for the question. So yeah, thank you again, Dayan, for a great presentation here. And so we will send a follow-up email after this with a recording of the webinar, so everyone can have that for the records. And if there's any, if you think of any additional questions, you feel free to respond to that email, and we can help do our best to answer this.
No problem, Ron. Thanks for the opportunity.
All right, take care, everyone. Have a good day.
Bye.
Bye-bye.